Welcome to this presentation on electromagnetic neurotechnologies. In this playlist, we will explore how electromagnetic fields intersect with neuroscience to revolutionize diagnostics and treatments. We'll break down the origins of electromagnetic neurotechnology, how the integration of electromagnetic science evolved, the concept of neuro-electromagnetism, understanding how electrical and magnetic fields are generated by brain activity, diagnostic and treatment applications, how these technologies are used for structural and functional brain imaging, as well as innovative therapies. We'll also touch on specific frequency ranges and cutting-edge tools. This field is vast, and its implications for healthcare and industry are transformative. Let's get started. The story of electromagnetic neurotechnology begins with two key discoveries. 1. Electromagnetic fields. In the 19th century, scientists like Michael Faraday and James Clerk Maxwell established the foundations of electromagnetism. Maxwell's equations showed how electric and magnetic fields interact. 2. Neuroscience advances. In the 20th century, researchers discovered that the brain operates using electrical signals. The work of pioneers like Hans Berger, who developed the first EEG, demonstrated that these signals could be detected and measured. Over time, scientists began integrating these fields. By applying electromagnetic principles to brain function, they created tools to visualize and influence brain activity. This led to the development of magnetic resonance imaging, MRI, for detailed brain scans, electroencephalography, EEG, for tracking brain waves, magnetic stimulation, TMS, for non-invasive brain stimulation. Today, electromagnetic neurotechnologies offer non-invasive ways to diagnose and treat brain disorders. They have applications in neurology, psychiatry, and even industrial diagnostics, such as evaluating concrete structures. Neuro-electromagnetism refers to the study of the brain's natural electrical and magnetic fields. Our brain's neurons communicate using electrical impulses. Whenever a neuron fires, it creates 1. Electrical fields. These fields can be detected by placing electrodes on the scalp, a technique known as electroencephalography. 2. Magnetic fields. These fields are much weaker, but can be measured with MEG, magnetoencephalography. Understanding neuroelectromagnetism allows us to map brain activity, identify which parts of the brain are active during different tasks, detect abnormalities in brain function, such as epilepsy or brain injuries, develop treatments, use magnetic and electrical fields to stimulate or inhibit brain activity, helping treat conditions like depression and Parkinson's disease. This field is critical for developing both diagnostic tools and therapeutic methods, understanding of how the brain functions. Diagnostic electromagnetic neurotechnologies fall into two main categories. 1. Structural imaging, focuses on visualizing the brain's anatomy. In this category, magnetic resonance imaging uses strong magnetic fields to generate detailed images of brain tissues. And microwave imaging uses wave to create permittivity profile of the brain materials. Also, computed tomography uses X-rays to capture cross-sectional images. 2. Functional imaging, tracks brain activity in real time. In passive technologies category, these simply measure brain activity without intervention. EEG and MEG are prime examples. EEG measures electrical activity on the scalp, MEG captures the magnetic fields generated by neuronal currents. In active technologies categories, these actively influence brain function. fMRI, functional MRI, measures changes in blood flow, active brain regions. Examples include transcranial magnetic stimulation, TMS, and deep brain stimulation, DBS. These technologies are essential for diagnosing conditions like epilepsy, brain tumors, and neurodegenerative diseases. Electromagnetic fields are not just for diagnostics, they are also used for treatments. Here are key examples. Electroconvulsive therapy, ECT, uses low-frequency electrical currents, around 50 Hz, to treat severe depression. It remains one of the most effective treatments for drug-resistant psychiatric. 1. Deep brain stimulation, DBS, involves implanting electrodes that deliver electrical pulses to specific brain areas. Frequencies typically range from 120 Hz to 200 Hz, it used for Parkinson's disease, essential tremor, and OCD. 2. Electromagnetic RF, radio frequency, and microwave frequencies, 10 MHz to 3 GHz, to heat and destroy cancer cells. The targeted heating sphere surrounding healthy tissue. 3. Temporal interference stimulation, uses two high-frequency electrical fields that intersect to create low-frequency stimulation. This technique could provide non-invasive DBS alternatives. Understanding these frequency ranges helps tailor treatments to specific conditions, making electromagnetic neurotechnologies powerful tools in modern medicine. In this part of the presentation, we'll discuss their principles, advantages, and limitations, providing a clear understanding of how these technologies are applied in clinical settings. We'll also explore the different methods of brain imaging using electromagnetic technologies, focusing on both structural and functional imaging. With structural imaging, the goal of structural imaging is to capture detailed images of the brain's physical structure. This is crucial for identifying anatomical abnormalities such as tumors, injuries, and malformations. The main technologies used for structural imaging include MRI, CT, and emerging techniques. The slide introduces two important concepts. 1. 3D resolution space. This graph shows the trade-offs between O time resolution, delta t in seconds, how quickly the technique captures brain activity. O spatial resolution, delta x in millimeters, how precisely activity. O contrast, delta c/c%, the ability to differentiate between different brain regions or activities. 2. Multidimensional acceptance space. This diagram highlights the factors influencing the usability of imaging technologies, such as operator dependence, training requirements, miniaturization, portability, cost, patient comfort, and harmlessness. Together, these concepts provide a framework for evaluating and choosing the right imaging technology for specific needs. In summary, the clinical scope of brain imaging encompasses a wide array of electromagnetic technologies for both structural imaging. These tools offer powerful insights into brain anatomy and activity, paving the way for better diagnosis, treatment, and understanding of the brain. Now, let's move on to structural imaging electromagnetic neurotechnologies comparison. Magnetic resonance imaging, MRI, works by using magnetically labeled water protons as tracers. When the brain is exposed to strong magnetic fields, these protons align and produce signals that are converted into detailed 3D. MRI offers high spatial resolution, making it ideal for detecting fine structural details. However, the temporal resolution is low, meaning it's not suited for real-time imaging. MRI is also expensive due to the sophisticated equipment involved. Computed tomography, CT, X-ray beam, to generate 3D images. Like MRI, CT scans provide high spatial resolution but have low temporal resolution. CT is particularly useful for detecting bone fractures and acute bleeding. However, it involves ionizing radiation, which can be a drawback compared to MRI. Microwave-based imaging is an emerging technology. This technique involves sensing temperature and conductivity changes using microwave antennas. Measuring signals reflected from tissue boundaries to generate images. Microwave imaging can detect temperature variations and blood volume change be converted to electrical signals for analysis. It offers high temporal resolution but still has unspecified spatial resolution. One advantage of microwave imaging is that it has the potential to be less expensive compared to MRI and CT, making it a promising tool for broader clinical use. Now, let's move on to functional imaging, which focuses on measuring brain activity in real time. Functional imaging is essential for understanding how different brain regions work and interact. It helps diagnose conditions like epilepsy, monitor cognitive functions, and guide brain surgeries. The primary modalities are EEG, MEG, fMRI, FNIR, and PET. Each method has distinct characteristics in terms of spatial and temporal resolution, invasiveness, and application. To summarize, these functional imaging techniques vary in their strengths and weaknesses. For example, EEG and MEG are ideal for high-speed recordings of brain activity but lack fine spatial detail. fMRI offers excellent spatial resolution but is slow and noisy. FNIR is portable and non-invasive, making it suitable for flexible clinical use. PET provides detailed metabolic information by its invasiveness and cost. Each technique is suited to different clinical and research applications, and often, combining multiple methods gives the best results. In conclusion, electromagnetic neurotechnologies represent a dynamic field with profound implications for medicine and industry. From diagnostics to treatments, these tools harness electromagnetic principles to unlock new possibilities for understanding and healing the brain. Thank you for joining this presentation.